Chapter 4, Section 19, Chemical Control of Microbial Growth. Now, we have quite a few chemicals and compounds that we use in your normal life, in the lab and everywhere, that we have developed for means to take care of all the germs, all the bacteria, all the fungi in our environments. Now, some of those chemicals and compounds are what we call killers. They will kill the vegetative cell, whether it's a bacteria or fungi or virus. We call these the cytals: bactericidal, fungicidal, virucidal. But some of the chemicals and compounds that we have in our cleaners and things like that do not directly kill by action. They inhibit growth. These are things that we call statics: bacteriostatic, fungostatic, virustatic. This kills, this inhibits, slows down. Why would you want to have one or another? Well, depends on just what you want to be exposed to. We've gone through and we've made lots of different chemicals and compounds for means of disinfecting or decontaminating, disinfecting and whatnot, sterilizing. Bacteriostatic, bactericidal, bacteriolytic. Lytic means lysis. This will cause the cell, the bacterial cell, to split open. Static inhibits, cidal kills, lytic lyses. This is what detergents do. Okay, really good, strong detergents go in and insert into the cytoplasmic membrane and cause the cytoplasmic membrane to come apart. Anything in the cytoplasm now gets leaked out. Cell dies. Bactericidal will damage enzymes, will damage membrane, will damage DNA, causing the cell to ultimately die, but it's not going to lyse open. Static is just going to slow down metabolic activity, inhibit an enzyme or two, stop something from happening for a while. So when you compare them side by side, red line is total cells. So you did one of those hemocytometer things. You counted total number of cells present, living, dead. Green is viable. How many are living? So you go. You add in. You allow things to grow, increasing in numbers, increasing in total cell, increasing in viable cell count. Boom. At this point, you add something. Everything plateaus. Given time, and then you remove that agent, numbers take off again. Notice nothing was forced to die here. Everything just kind of went and maintained numbers. Bactericidal, on the other hand, you allow things to grow, total cell count, viable cell count, and then you add in your bactericidal compound. The total number of viable cells, living cells, will plummet. Boom. Total cells going to stay there. Because remember, this is living and dead. This is just living. And then the bacteriolytic, well, everything goes up, everything goes down, because the bacteriolytic is making everybody break in half, split open. So you have no total cells, living and dead, left to count. When we develop something new, okay, ooh, look, it kills, it damages, we need to have a way to compare it to pre-existing. How's this working? What's it doing? One of the ways we have to characterize, so we can compare apples to apples, is what is called the minimum inhibitory concentration, MIC. What is the smallest amount, the smallest concentration of your new agent, your new chemical, your new compound? What's the smallest amount so that we can see an inhibition of growth, whether it's killing or it's acting statically? All right. We can do this either in liquid medium, or we can do it on an agar plate using what's called disc diffusion. So if you're going to do the broth means of doing this, to find out the minimum inhibitory concentration, you're first going to take some broth that you have your chemical, your compound, your whatever in, and then you're going to run a series of dilutions. Boom, 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 boom. High concentration, low concentration. And then you're going to inoculate every tube with the same number of bacteria. And then you're going to step back. And you're going to wait. And you're going to see what grows. At high concentrations of your new chemical, your new compound, nothing grows. At low concentration, everything grows. But there's going to be a tube where it goes from no growth to some growth. That tube, that last tube of no growth, whatever that dilution is, that concentration is your minimum inhibitory. Anything below that, it doesn't stop. Anything above that, it's just a waste of your compound. We can also study this using agar plates. What you do is you take your bacteria of choice, you spread it out on the surface. What you're shooting for is so many bacteria on this surface that instead of individual colonies like we saw with the pour plate or the spread plate, now you have so many colonies that they're growing on top of each other, and it's now going to look like one giant colony. They call that a lawn of bacteria. But before the bacteria can grow, you're going to put paper or filtered discs on there. Each of these different colored filters you see here has been soaked in a different concentration of your compound. As this plate is incubated overnight, or for 1 to 2 days, as the bacteria colonies grow to the point that they're now just one giant growth, a lawn of bacteria, your chemical, your compound of interest, has diffused out of those discs. Higher concentrations have inhibited growth at a greater diffusion distance than the lower concentrations. They call these the zones of inhibition. It's basically a zone of death. Your chemical, your compound, your antibiotic, your whatever, is at such high concentration, any microbe that is close to that disc is overwhelmed and killed. At this concentration, well, there's just not enough there to really inhibit anything, and the bacteria basically grow up and touch. This one and that one, the concentrations there were so low that the bacteria are able to grow closer and closer to the disc. What you then do is there are certain tables for every known chemical compound. You go and you measure the diameter of that, and it'll tell you whether or not this bacteria is susceptible or resistant to your chemical, your compound at that concentration. We go through and then we classify. Once you have something new and you showed, hey, it works, here's the minimum inhibitory concentration. You know, we have things that are sterilants, disinfectants, sanitizers, antiseptics. Disinfectants and antiseptics sound a whole lot alike. Disinfectants, though, are on surfaces, tables, cutting boards. Antiseptics are on living tissues. So antiseptic hand wash or antiseptic sanitizers you have, or antiseptic washes that, you know, like if you're going into surgery and they rub that stuff, that orange stuff on your body, that's an antiseptic. It's killing stuff on the surface of your skin. Your skin's living tissue. If you're wiping down a surface because you're going to have to do something there and you want to get it somewhat clean, that's a disinfectant. The alcohol you're using, it's going to be more in lab to, clean the tables. That's going to be more disinfectant than sanitizers. Sanitizers, less harsh, reduced numbers, but don't really kill that many. Disinfecting with bleach, disinfecting with alcohol, kills most microbes, vegetative cells, but does absolutely nothing to endospores. If you kill all vegetative cells, you irreversibly damage all spores, endospores, so that they cannot germinate. That's a sterilant. So you go through alcohol. It's a great antiseptic if you pour it on a wound. You pour it on your skin. That's why if you go and you look at most hand sanitizers, what they call hand sanitizers, there's going to be alcohol in them. You go into the hospital, you're going to see that phenol is going to be one of their antiseptics of choice in the soaps, in stuff that they're going to rub on you right before you go into surgery. Hydrogen peroxide is an oxidizing agent. It's a great antiseptic. That oxidizing, remember, H2O2 breaks down into water and the singlet oxygen, which is going to go through and strip off hydrogens off of anything and everything. Betadine. Ow, that stuff I still remember as a kid getting that stuff rubbed on. It's an iodine compound. Great antiseptic. Sterilants, disinfectants, alcohols, chlorine gas. Yeah, we use chlorine gas. The same stuff they used in World War I that they, pumped into, all the trenches. Okay, we use it nowadays as a disinfectant for drinking water and water treatment plants as we get it ready to go to, municipal water systems. So we use chlorine or chlorine compounds. That's what's in, pool water. That's why it smells, chlorine. Sodium hypochlorite, bleach. Copper sulfate. Yeah, copper sulfate is actually a great algicide, and they're starting to see it's also a great bactericidal properties. Formaldehyde, the reason that works great is because it's an alkylating agent. Goes through and acts as a disinfectant, a sterilant. Formaldehyde and glutaraldehydes irreversibly cross-link disulfide bonds, of neighboring proteins. Not just the disulfide bonds holding the tertiary structure of a protein, but neighboring proteins will now get irreversibly bonded together. Acts as a great sterilant. That's why they used formaldehyde in embalming fluid, to keep the bodies from rotting. It killed most things there. It sterilized, at least disinfected most of the interior. Didn't do much to the gastrointestinal tract. They didn't always flush those with embalming fluid. Hydrogen peroxide, I already talked about. Ozone. Ozone's great. Ozone, O3. Water disinfectant. Pine oils, pine sol. Protein denaturant. Yeah, pine sol. That stuff your grandma used because it made the house smell like pine trees and whatnot when she used to clean the house. Yeah, there's a reason. It's a protein denaturant. It's an all-purpose disinfectant. Doesn't kill a lot, but it disinfects. It brings the numbers down below a certain level so that it's not too bad for you.